everyone. We're going to go ahead and get started today. Thank you. It's about noon right now. So thank you all for coming out today for a Rough Cut Seminar Series from the Institute on Ecosystems, especially on this snowy day. But I have the pleasure of introducing Dr. Brian Smithers, who will be presenting today. And Brian is an assistant research professor in the Department of Ecology here at MSU, as well as the executive director of GLORIA Great Basin, a nonprofit organization that puts together groups of citizen scientists and agency personnel to monitor the long-term response of mountaintop plant communities to climate change. And GLORIA Great Basin monitors 29 peaks throughout the Great Basin region and is part of the International GLORIA Project, which you might have to explain what the acronym means for that. But that monitors hundreds of mountain ranges all over the world. And Brian is in his second year at MSU with research that generally focuses on the interspecific interactions of plant species as they adjust their ranges in response to climate change. So let's give a round of applause for Brian.
Thanks, Madison, and thanks, Bruce, for inviting me to give this talk. I'm pretty excited to share some of this stuff that's just coming out. Our first paper actually from that GLORIA network, which I'll talk about in a minute, is due any day now to hit the presses. So that's partly what I'll be talking about today. So the question we ask, we're all asking, or a lot of us are asking, is what's going to happen to our species in response to climate change, or what is happening to our species in response to climate change? I'm sorry. What we're asking is louder, that, Brian. Louder. I'm sorry. Sorry. I kind of deserve it. Where our species will go. I like to stand on top of pretty mountains, and so for me, I want to know where alpine plants are going to go. And there's some really good reasons why we might want to focus on alpine plants as a proxy to look at where species are moving or going to move in general. When we just generally look at a mountain, it's just to bring it all the way back to the basics here. We can generally group our plants communities into zones, right? We going from low down to higher up. Some of my undergraduates are here from Rocky Mountain Vegetation. We do this a lot. We talk about what kind of habitat we might expect, or what kind of species we might expect here versus somewhere in mid elevations. And these are generally, we've known this for a long time, generally representative of climatic conditions, often edaphic conditions as well, but that we can group species based on a certain set of factors, generally on the side of a mountain, and generally that'll hold true. And we might expect with climate change that there'll be some reshuffling of that. How that happens, though, I think there's still some debate. Will the species that make up the foothill woodland community just move up in lockstep? Will individual species move differently? With respect to alpine, oh, sorry, hang on. And going back, this is fun, by the way. I'm going to write on this, and you get to see it. We're going to be focusing on this, on these communities at the very top, and there's really good reason for that. There's of course concern that if you notice the chaparral community, if it wants to move upslope in response to warming, it can just move on up, right? But where do these guys go? There's nowhere to move up to. And so there's specific concern for some of these species. Polar bears, right? Where are they going to move north to? Alpine plants, where are they going to move up to? Alpine plant communities also have some nice characteristics concerning whether they're good proxies for how we think about them in terms of climate change. Maybe more so than many species, alpine plants are really coupled to the environment. They're really coupled to climate. There's not a whole lot separating this plant from the air temperature. Trees can moderate, can modulate environment in many ways. Like the canopy can modulate what's happening below environmentally and can buffer a lot of these conditions, but alpine plants are really in it. And so because of that, what we see on the landscape really is a function of temperature, precipitation, snowpack, these very basic climatic parameters. It also happens that alpine plant communities are generally pretty low on the disturbance level. once you get above a certain elevation, you're not typically talking about much human disturbance or some legacy effects from sheep grazing, but it's pretty minor relative to a lot of systems. Not only that, but there's limited climatic or environmental disturbance that happens on a mountaintop. Even glaciation typically is affecting things further downslope from the actual mountain. And so we've got some really undisturbed areas with the plant communities found there, and that are really tightly coupled to the environment. So they're a really nice harbinger of biotic impacts of a changing climate. Now, when you talk about a species or a community that's going to respond to climate change, we can't expect things to just move. You know, it warms up and so things move. There are a number of lags associated with this. This paper by Alexander et al. In 2017 highlighted 3 of these lags: dispersal lag, an establishment lag, and then an extinction lag. So here we're talking about a change in community or change in species. A species may be sitting where it's warmer than it would like, but it's there, and so it's going to stay there. It's obviously trying to get its young somewhere else, presumably upslope, but it's going to face a number of challenges in order to do that. First of all, can it get its seeds to that higher elevation? These are color coded. I've got them here. The orange here are demographic or physiological issues. The green are biotic interactions, and the blue are physical issues with the physical environment. And so you can see they face a whole host of things that might either help dispersal or might hinder dispersal. So there's going to be a lag there. Now, even if they can get their seeds to that new spot, that new climatically suitable spot, they've got a whole nother suite of issues they need to deal with in order to get established. One being that there are already species there who are still maybe in their own dispersal lag. So now they're competing with other species who are already there. It could be missing a mutualist, a mycorrhizal mutualist perhaps, that end up with a further establishment lag. And then in order for this community to actually change from what was there before to what is coming or what is there now, the species who were there to begin with have to go extinct, and that's its own lag. That also takes a certain amount of time. And so this can really change how these species are moving around the landscape. It turns out that, given this, there's a good amount of data, or there's at least one bit of data in a very large data set to show that species richness has increased in some of these mountaintop systems. There is a paper that came out just last year looking at mountaintop alpine plants. You probably can't see this in the back there, but it's this is just a map, latitude and longitude here. But looking at each one of these, it's highlighting one of the mountain areas. And it's just going from 1900 to 2000. So they had a data set from 1900 looking at the alpine plant communities on these mountaintops, and they resurveyed these in 2000. And along the y-axis is richness. And across the board, there's just an increase in species on mountaintops all over Europe. I think this was universal in their analysis, that every single one of their peaks showed an increase in plant species richness. And so probably what that is we've got species that have gotten through their dispersal lag and their establishment lag, but we're still waiting on the extinction lag. So species have moved up. The species who were there haven't gone extinct yet or won't, right? So it looks like dispersal and establishment are likely a little bit faster than extinction or faster, according to that data. Some assumptions of this, though, are, first of all, climate's warming. It's a pretty fair assumption at this point, and they make sure they make that clear in the paper that the climate has warmed all over Europe. But it's also, we're assuming then, if this is happening in response to climate change, in response to warming, we're assuming that the areas upslope are in fact cooler and or wetter than the areas downslope. Another assumption. They're assuming species can move to those cooler and wetter areas, and that climate structures where species are found. Those might seem like pretty intuitive, unautomatic assumptions for you to make, but they're not. it's not necessarily true that areas upslope are always cooler or wetter. Some species can move better than others. It's not necessarily true that climate warms, the climate structures where species are found. In fact, this debate goes way back to some of the foundations of ecology. Frederick Clements, of course, believed that climate absolutely structured where species are found, and that you basically, if you knew the climatic parameters of an area, you could predict which species you ought to find there. Henry Gleason did not see that way. In fact, he believed that species were individualistic in how they responded to environmental parameters. If you're Frederick Clements, this is what you might expect to see. If each one of these lines is a species, we've got some environmental gradient here. Let's just call it temperature. You would expect to see, according to his climax theory, you would expect to see basically these communities. So we've got 1, 2, 3, 4 species in that community, separated by these ecotone barriers. And then you get another set of species after that. And then if temperature warms or something happens, these would all just move together. He didn't say that, but that's the natural extension of that thought process. Gleason didn't really buy that, and he thought the world looked more like this. So these are both real data sets, by the way, and so both of them are right, unfortunately. Where each of these lines is a species, and you can see there's no community structure to this. Each species is responding in its own way to, in this instance, moisture. So that jury's still out. Henry Gleason, by the way, he came out with this theory. He thought he was going to change ecology, and then he was totally panned. And so he's like, screw this. He quit ecology and just became a taxonomist, a plant taxonomist. He's like, people aren't going to, pan plant taxonomy. So he got out of the game. And Clements' theory persisted for a long time. Now, so taking these ideas, we've got, this long-standing debate. Are plant communities structured deterministically, meaning it's just all about the environment, it's all about the climate. Provide a climate, you're going to get a certain set of species. Or is there some stochasticity to this? Is it more random than that? That was another, I failed to mention that. Henry Gleason was not only individualistic, but that a lot of what happens on a landscape is happening randomly. Whatever species get there, that's your community. So we wanted to ask some questions pertaining to alpine plant communities. Let's say you've got a mountain range here. It's just represented by these 3 mountains of various height. And you've got some sort of regional species pool represented up here at the top. Just keeping it simple, I've got 6 species on here. And I've color-coded this, which isn't that important, but just so you know with the colors that you're looking at there, where the blue are the cool adapted species and the redder ones, or the red boxes, are the warm adapted species. We wanted to ask some questions pertaining to plant communities to try to predict where they might go based on some of the things we just talked about. We came up with these 3 hypotheses. This first one I'm going to mention to you, and then I'm not going to mention again. But the first one was that elevation, and here we're using, for elevation, it's really a proxy for temperature. Because that's true at broad scales. We know that adiabatic lapse rates hold at broad scales. So generally, elevation, temperature, explains more variation in species composition within each summit than across the mountain range. Okay, that's all I'm going to say about that for right now. If you want to ask questions about that, we can talk later. That was our first hypothesis. Our second one, which I do want to talk more about, is that elevation, once again, being a proxy for climatic conditions, elevation explains more variation in climatic niche affinities across the mountain range than within each summit. I'm going to talk a lot about what these climatic niche affinities are, so don't get hung up on that just yet. Essentially, what we're saying here is that elevation explains more variation in climate across the mountain range than within each summit. And I'll explain how we're measuring climate, because I think that's actually the interesting part here. But that we would expect more variation across the mountain range than we would within each summit. And then a third hypothesis is that if you've got increasing, and let's just once again call this climate or climatic range, you've got increasing climatic range in an area, you would expect increasing community turnover. Folks in plant circles know this well. If you've got increasing microclimatic range, you get higher beta diversity. You get higher species turnover because you've got more habitat change. Okay, we'll revisit these hypotheses as we go, so don't memorize. As Madison mentioned, this data was collected as part of this international protocol/effort called GLORIA, which has got to be the worst acronym that's ever been created. Watch this. The GL come from the first word. The O and the R are okay. The I is okay. We're missing an I there. And then A, and then there's no E. So environments goes away. But anyway, that's the network. It was named by Austrians, and hopefully this works better in German. It was established in 2001 in Austria as a way to do long-term monitoring on mountaintops cheaply, so that it could be done all over the world. And it really has taken off. It is now all over the world, with the exclusion of Antarctica. Hundreds of peaks are being monitored typically every 5 years, is the monitoring protocol on that. Peaks going from the tropics in East Africa all the way up to some of our polar regions. And some, to date, no worldwide syntheses have been done on this. It'd be a monumental undertaking, but certainly one of the papers I just showed was a European synthesis of this. So we're using this protocol that monitors these peaks. We formed a group, and we, as the royal we here, I was not around yet in this case, but in 2004, started monitoring peaks in the Western United States, formed a nonprofit organization a couple of years ago called ourselves GLORIA Great Basin, where we monitor this set of peaks across the Great Basin, California and Nevada. 8 regions, 29 peaks. I'm going to mention this here because I think this is maybe the coolest part of what I do is we have no money, and so we do everything based on volunteerism, and we get an amazing response. Every year, I'm now turning people away to pay us to climb peaks and then be put to work. We get this great mix of agency personnel, citizen scientists. We work with nonprofit organizations like Native Plant Societies, and just have the best time doing this. Just maybe another model for how research can get done is by using people, because we've found that people want to be involved as much as possible. Most of these people are expert botanists, and so they're very useful to us. And so here's just some folks helping out. We invented a new kind of survey within the GLORIA protocol because these protocols were created in pretty wet Europe, where they could put down a 1 meter plot and get all kinds of data. If we put a 1 meter plot down on that, we find out it's covered with rock, and we can do that repeatedly. We can continue we get great repeatability with that. So we've created a couple of different protocols. One of them we call a downslope survey, which is where we run from the top of a peak, or roughly the top of the peak, down to the subalpine zone, a series of these 100-meter long transects. And in each of those, we divide them up into 10, I don't know if I'm doing 10 here or not, but you get the idea, 10 sub-transects. In each of those, we collect data, we collect a belt transect, essentially. It's a meter wide, and in that we census everything that's in there. And then we go back through and we use this handy tool to measure frequency, where we're just doing point counts. We work at 13,000 feet, so things get a little funny. This is a 2-pronged pointer, and if a trident has 3 prongs, what do you think we call this thing? A bident, which I just learned is actually a word. We thought we made it up. And if the Vice President of the United States shares that name, what do you call it? We call it a Joe Biden. Yeah. We love our Joe Biden, which predates the current election stream. We're keeping the name. So that's our Joe Biden, which we use to get frequency counts within each of these 10-meter segments. We've got these now running on 5 different peaks just in one mountain range, the White Mountains. So 5 peaks there, going from high to low, roughly. White Mountain Peak, Sheep Mountain, no, that's Barcroft, Sheep Mountain, etc., on down to Campedo. Typically 8 to 14 of these 100-meter belt transects, depending on the height of the mountain, essentially. The White Mountains, just to point out where we are here in the map of California, if you haven't been to this area, the White Mountains are sort of a standalone mountain range just to the east of the Sierra Nevada, and just to the east of the highest points of the Sierra Nevada. So a lot of the moisture gets knocked down by the Sierra Nevada before it gets to the White Mountains. It's a very dry mountain range, as you can see. They're very high themselves. The highest peak in the range is over 14,000 feet, 14,235 feet, to be exact. You may have heard of the White Mountains because their claim to fame are these trees, the bristlecone pines, the Great Basin bristlecone pines, one of which in the White Mountains is the oldest known non-clonal organism on Earth, a tree called Methuselah. But we literally look down on the bristlecone pines from our work. So I mentioned briefly that how we measure climate is probably the neat part of this study. We use what are called climatic niche means to look at to use the species that are there to actually tell us what the climate is. We find that if you try to put temperature sensors, go out every couple of weeks and sync soil moisture probes, you get these snapshots that tell you a bit about the climate, but probably don't tell you what the plants are actually experiencing. However, the plants know damn well what they're experiencing. And so if you can find out more about that plant's climatic tolerances, you can infer a lot about the climate of that area. So I'm going to give you some definitions here. The CNM, you're going to see a lot, the climatic niche mean. This is the mean climate where individuals of a species are found. If you can take a species and you've got records of it all over California, and this is in fact the case, there are records of collections all over California that are now housed in the Consortium of California Herbaria that you can search. And so for any given species, you can get all of its location records all over the state of California. Then what you can do, you slap those on a map, then you can overlay that with, pick your favorite climate layers in Prism, for example, and you can say, all right, what's the average temperature in January for all the places where you have that species? What's the mean annual precipitation? Pick your climatic variable. And so we do that with, it just so happens that a paper came out where they took all of those herbaria records, cleaned them all up. They're all for every species in California. And so we have this great data set of locations for each species everywhere in California. And you can take that, and then for each species, I'm going to go back. Let's say we've got a species, say this is Elamis elamoides, a species of grass. We can say, where do we find it in California? We plot it on this map, and what we're looking at here, I believe, is some sort of precipitation map underneath it. So we can extract the precipitation values from each of the raster grid cells from each of these locations. And what we find, for example, this is climatic water deficit. These are mean values of climatic water deficit for just a couple of species, and this works pretty well when we look at things in the literature of what we might expect Calaria macrantha can tolerate. These turn out to look pretty good, and so we get a good spread of species that are more adapted to wetter areas. Drabelithosperma has a low CWD value. Calaria macrantha has a high CWD value. When we get those means, you just see kind of a scatter shot there. So we can do that for each species. So for each species, then we can, once again, we can do this for the mean temperature in July if we wanted to, or pick your variable. For each species, we can then scale that up and use those to calculate some community metrics. we call that a CCNM, a community climate niche mean. That's the C right there. And the way we do this is from our sampling protocol, is to weight this based on the frequencies that we collected with our GeoVident. So we're walking along, let's say this transect here that we're looking at there is one of these transects here. We can say, let's say just for the sake of simplicity, that we got 3 species in this. We'll call this A, B, and triangles are C there. We can then take each of those species that we found in that transect, put them on our map of California moisture, and extract the values from that. And let's say for each species, we got these means. There, yep. Let's say our blue species, our species A, had a mean CWD value, climatic water deficit value 300, which kind of makes sense. Looks like it's up in here in a cooler, wetter area. Our square 700 down here in the Sonoran Desert. That makes sense. That it would be a higher CWD value. And we want to calculate some metrics for this community. We can do some pretty simple math. Hey, simple if the computer cooperates. There we go. So we can take for species A, 300. We got 3 of those. Plus species B, which was 700, right? Oops. And we had 2 of those. Plus species C, we had 500. And we had 1 of those. You do that math, you get a value of 2,800. Divide by the number of species, and you get a mean value for the CWD of this community of 476. You've used the species which are present in that transect, in that community, to essentially tell you the climatic water deficit of that spot, of that transect. Once again, and you can do this for a number of variables. Let's check my math there. Oh, good. And that's what we did for each of these transects, each of those 10-meter sub-transects within those transects, to tell us what's the CWD here, 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 et cetera, all the way up and down the mountain. And get a really fine-grained analysis of the climatic conditions in each of these areas. All right, so let's get back to hypothesis 2. Elevation explains more variation in climatic niche affinities across the mountain range than within each transect. So these climate niche affinities are what we were just talking about with those community metrics. So we expected that elevation would explain more variation in those across the mountain range than within each summit. And what do we find? What we're looking at here is elevation along the x-axis. We've got, I'll just stick with that for a minute. Just look at this one with me for a minute here. On this graph, we've got a series of colors. Each color scheme here represents one of the peaks, and each point is one of the transects on that peak. And so actually go back to, because we were talking about water deficit, if we just ignore peak for a minute and just look at the mountain range wide response here, it's super clear. We have a very strong relationship between elevation and climatic water deficit. As you move up slope, it gets cooler and wetter. That's kind of a theme, right? We know that on broad scales. But if you zoom in and try to ignore that black line for a minute and look at some of these peaks, individual peaks, what you find is that relationship really breaks down. It even goes positive in some situations. For example, this is a Campedo peak, one of our lower peaks. Our lines going this way, the dashed lines represent on this insignificant values. They're insignificant relationships. We did this for climatic water deficit, for mean annual precipitation, mean maximum temperatures of July, mean minimum temperatures of January, and that relationship continues to hold. We have these strong relationships across the mountain range that break down at the peak level, at the peak scale. Okay, think about that for a minute, or hold that thought for a minute. And I want to jump to our third hypothesis. If you looked at a more local level, so at the scale of a peak and then even at the scale of a transect, we would expect if you have increasing climatic niche affinity range. So let's just say, let's use that climatic water deficit again. If you have a large range in climatic water deficit across this transect, we would expect to see increasing turnover in species composition. We expect to see from from here to here. If their range is larger, we would expect to see greater species turnover between them at both this scale and at the scale of each of our 10 meters. And we do see that, at least for some of our climate metrics. Once again, dashed is insignificant, solid is significant. But we see that from just going for, that on any one transect, each of the points here is one transect. The range of, for example, here the range of July T max CCNM. So the range within that transect, the range of maximum July temperatures, as that increases, our beta diversity increases, our species turnover increases. So as we get higher range, we get different species there. What does this mean? The way we interpret this is a couple of different ways. First of all, species turnover at broad scales is less climatically driven than at local scales, meaning, and that's that first hypothesis which we didn't actually spend time on, but that essentially there's luck involved. There is that Gleason is at least partly right. Jumping to the bottom two here. Environmental sorting is stronger at broad scales. That was this here. At broad scales, at the scale of the mountain range, the environment has a lot to say about what's going on, but not so at smaller scales. And that third hypothesis, microclimate and site-specific factors are stronger at smaller scales. We find that, as our range increases here, as there is an increase in microclimate variation along a transect, we get greater species turnover, and that this is strongest at smaller scales, where we have, when we're at the microclimate scale. And that scale and microclimate really then matter to where species can move to keep pace with climate change. What this is really saying is that in some places, there's quite a lot of climatic variation just along one transect. In fact, we found that there was as much climatic variation along one transect at one elevation than we found between that transect and 100 meters upslope of that. You follow that? There's so much microclimatic variation at one elevation that it's the equivalent of moving up 100 meters in elevation, which is in some cases the entirety of our transect range. What that likely means is that species don't necessarily have to move up slope to keep pace with climate change. Not necessarily, right? At least in some places where there is higher microclimatic diversity, species can move around the corner, can move behind that rock over there, can get down into that swale over here that's got just a little bit cooler temperature. There's that much habitat variability on the landscape in alpine systems. This is a rare occurrence of maybe some, I won't call it optimism, but not so bad. It may not be so bad. We may not just wholesale lose all of our alpine plant species, that there is quite a bit of variability on the landscape that may provide refugia for these species. I love this tool. I'm using it in a couple of other projects now. We've got a project going on the west slope of Sierra Nevada, the Tea Kettle Experimental Forest, where we're doing combinations of thinning and burning and not thinning to look at some different management protocols and how we might try to fix our fire problem in the West. And one of the ways that we are now looking at, we're about to burn in the fall, we're about to burn 500-acre area based on one of our favorite thinning and burning strategies. And so we're going to be using this the CCNM, the Community Climatic Niche Mean methods, to look at how things are changing with the fire, and then how they change over time. You know, how do the climatic parameters look right after you burn versus 5 years down the road? How does that look compared to areas where you thinned and burned versus just burned? And so this tool, I think, has got a lot of potential. I'm really excited about it. Host of folks to thank. We've some funders, which is great. I'm thanking myself essentially with very great basin. But what I'm really thanking are the volunteers, and it's a host of people who make this happen every year. Volunteers. Really is the way it's happening. So thank you. That's all I've got for now, and I'd love to have a little discussion if you've got some time.
Question, there are microphones along the benches, so you could turn it on before you ask your question. That'd be great.
Did you turn your microphone on?
A little green.
Excellent presentation, Brian.
Thanks.
Do you think that any of the signals of the variability at the different scales reflect the dynamism and the vicariance that's kind of really strong on the Great Basin?
What do you mean? What's giving your vicariance?
So the chance events of species arriving,
the
island biogeography.
Oh, sure.
Like, part of it seems to me that there's so much diversity across mountain ranges out there.
Yeah, we wanted to do this. Scale is obviously super important in what we did here. And we really had hoped to do this, go to the next level, mountain range to mountain range. And we just didn't have the data ready to do that yet. There is, just as and Eric knows the Great Basin far better than I do, there's unbelievable species turnover from mountain range to mountain range, and it's kind of a unique situation. It's 300 and something mountain ranges in Nevada, and they're all sort of lined up, and they're separated by these broad valleys. And so there is this wonderful vicariance situation happening among the mountain ranges, and I wish we had been able to do that analysis. Stay tuned, I think is the answer there. Yeah, question, thanks. Yeah, Scott.
Thanks, Brian. I just wanted to make sure I understood something about the ordinarian collection itself, because it's so foundational to the calculation of that metric. How, to what extent does that represent the true distribution of the species?
Yeah, and before anybody asks this, I'll just say it. We only have records for California. A lot of these species ranges go all over the western United States, up into Canada and into Mexico. So it's a subset. And we know we're limited in that way. We're also limited to where it was collected. And we have much better records for some species than others. Species that are widespread or common are much better represented. Species that are rarer are less represented, but maybe that's accurate. They're less represented because they're only found in one or two spots. And then so if you have those two spots represented, you've got your range of variation essentially. It can be very narrow. It's a concern. It's a concern. We spent a lot of time looking at our values to make sure they made sense species-wise. And so I think we just have to call it a pretty good approximation of the species climatic tolerances or species climatic range. And then we're using those then to tell us about the community. And I think it's still useful, even though there are certainly limitations. Yeah, does that?
Yeah.
Yeah. Thanks.
So I'm curious about the, this is a really interesting and it's fundamentally about abiotic constraints on species distribution.
Right.
What about the biotic constraints and interactions with pollinators or whatever's dispersing this? Could that play a role, or how important is it to consider?
Yeah, I think it really certainly that your example there with dispersal or just with biotic interactions I think that really depends on on whether we're right about microclimate being so important. If there's very good microclimate habitat available nearby, those become less important. But they are super important. They're clearly important. Almost all of my other research that I do specifically looks at these biotic interactions. I'm finding I do a lot of work on tree-lined trees in the same system, and we know that the climate is perfectly acceptable for bristlecone pine and limber pine to be hundreds of meters upslope, and they're not. And it appears to largely be a dispersal and/or biotic mycorrhizal issue that's not allowing them to move. Yeah, it's big, and then at the same time, competition is not as far as we think, competition is not a big driver. It's not moisture limited generally. Once you get established, it's not anyway. There's plenty of moisture, this far below the surface, and it's pretty sparse generally. And so competition is probably not as big of a deal as it is in other systems, but I think the facilitation part is key. Yeah. Thanks, Mike. Good question, Anthony.
Could you go back to your one figure showing? Just tell me. I just want to make sure I understand showing the range of CCM. Yeah.
Yep,
okay. So each point. So I'm looking down at the one you highlighted, the range of July Tmax.
Yes.
So each point is a transect, and then those are across many mountain ranges.
Yes.
So you have some mountain ranges. So how comparable are those as far as the species that are there? I guess is what I'm trying to.
Yeah, that's a good question. So not. So I mean, I think they are comparable. They're not comparable because the species are the same. The 5 mountain ranges represent 3 different soil types. The species are actually quite different. However, so each of these dots here is representing one of the transects with the 10 segments.
And then you have a ladder rung pattern going up in elevation.
Yeah, and so and so we've got, this is another transect as you're going up.
And so all of those are 1 point, or each one of the lines?
This is 1 point here. Okay. That's another point there. So on this, so let's say this transect corresponds to that point right there. The range is we've got a T max for this section, and then another T max for this section, et cetera, et cetera. So we've got 10 T maxes. The range is just the difference between the highest and the lowest of those T maxes.
So on one stretch, you have considerable range, which is surprising to me.
Yep.
In maximum July temperatures.
Yes.
Would seemingly be a very small area comparatively.
Yes.
To the whole mountainside.
4 degrees Celsius. So being is a huge range, right? Now remember, these aren't actual values. These are extracted values from a climatic community niche.
So as far as the plants that are growing on that transect are concerned, there's that range there's that mean range in T.
Yes. Right. Now, once again, it may not be that there actually is that. The species that are there are saying that, but it could also be that that's a drier spot, and then so the species show up. The species that are found there can handle drier. They can also handle warmer, and so it may not be. Maybe it's 1 degree Celsius warmer, but a whole lot drier. Yeah, great question.
Just trying to make sure.
I'm glad I could answer it. More importantly,
How
hi Matt.
Beta diversity within a single transect? How are we getting Y axis?
Beta diversity is the species turnover from A to B to C, essentially. So each of those segments, it's the species turnover from one of those to the other.
It's an average.
And it's an average. Yeah, sorry. And then it's an average of those, right? Yes, sir.
So I'm thinking of that one thing that's missing from your assessment that I'm just curious about what you thought about is linking the life history with climate patterns. And that seems like if you chose a subset of species, let's say that have very different life histories, would they start to account for some of the variation that you see in your turnover? but how those have to link in some way with your climatic, just using the climatic mean, come up with some sort of matching of that life history or chronology, let's say, to precipitation changes, for example.
Right.
One of the things that strikes me as one of the major patterns that we're seeing in our region is shift to very wet spring, pretty wet fall, super dry summer.
Right.
And that species life histories will start to match to that.
Right.
I think.
Yeah.
But I just wonder if that would help explain, especially as you moved up in elevation, that might be a way to pick up some of the variation, maybe disguising what these differences in distribution.
Absolutely. And I don't know if what I'm going to say is in response to what you just said. I'm going to say what I think. The one part of this that hopefully somebody picked up on, it's one snapshot. This was one survey. I mean, over 3 years because it's multiple peaks, but it's one snapshot, and we're just matching things up. We do have 3 years of this, the 3 cycles of this data, and so what's coming next, the paper we're working on right now, is actually following this through time, and then we're going to start including real climatic parameters to try to drill into some of those questions. We don't have great climatic data. We weren't at the outset. We weren't measuring climate at all. We now at least have temperature sensors at one spot in each transect, but in a 100-meter transect, we just saw that the range can be quite big. We're having a hard time getting accurate or precise enough climate data to make those inferences. But we're excited about what's coming next with looking over periods of time and including the climatic information that we do have. Yeah.
On your previous graph, I think.
Yep.
But that one, yeah. I was wondering if you looked at the relationships within a peak, and if the greater microclimatic variation that existed within a peak, did that improve those relationships? Because you had said on for this slide that the overall relationship was really strong, but then it kind of fell apart at the peak level. So I don't know if you looked at that, and did it depend on how variable the microclimate was?
So if it were, I'm just trying to think through this here. If it were more variable, yeah, we've got just a mean value here for each transect. In the next slide, the next graph, we've got it kind of more expanded, right? Looking at the range. I wouldn't expect, like, I'm trying to think about those two together. Along an elevation gradient, I don't think elevation would have anything to do with whether there's more or less microclimate along any one transect. Does that make sense? I think that's a pretty random occurrence, or according to our sampling design anyway, that you run into an area that just happens to have a lot of nooks and crannies. That's going to increase your climatic range on that one transect. So I wouldn't think so. I wouldn't think the range. In fact, I think that would just kind of be scattershot on this graph if you had range on the y-axis.
Okay.
And
yeah, I
then
guess I
elevation
was
on
just
the
wondering
x-axis.
if you found any of those within peak relationships to be significant.
Right. That's hypothesis 1, which I didn't really talk about. But what we did find is that community composition was quite different. We found part of what Anthony was actually asking about was that there seemed to be a certain amount of stochasticity involved with, on a peak, you've got a certain set of group of species, on another peak, you've got another group of species that you're generally working with. I think a lot of that has to do with as I mentioned, soil issues, just different soil types. But even ones that are the same are working with a different set of species, and so slightly different set of species. Yeah, sorry, I think I'm straying from your question now.
That's okay, thanks.
Yeah, okay,
Yeah.
right. Just keep talking until they.
What years did you collect this data?
What years?
Yeah.
2011, 12, and 13. And then we just had another round that ended in 2018, so 18, 17, and 16, 5 years apart. And so those two sets of data, Tyler's entering right now.
Tyler.
Travis.
Jacob.
Who's Tyler? Jacob Crooks. Jacob, sorry, Jacob, is entering right now. So thank you, Jacob. Another volunteer to add to the thank yous. So yeah, we're excited to see that data.
So you mentioned you have visited several of these peaks in additional years.
Yep.
And we talked a little bit in person about how you've seen a lot of turnover year to year for a particular
Yes.
summit. How do you expect that to change your climate niche space? Because if you have complete turnover of your species diversity, then it might drastically shift that specific summit.
Part of the reason we stepped back from doing that in this first paper. So what you're getting at there is we go out 2012, and 2012 happens to be a really wet and cool year. And we go back 5 years later, we're there for literally one day in a year that's bone dry. We're going to see a very different response, probably. So there are a couple things there. Most of the species we're dealing with are long-lived perennials. And our botany, I think, is to the point where we do some pretty good forensic botany. Crispy dried things, we feel pretty good about still being able to identify. But there's certainly annuals, 2 species of annual, that would certainly be affected by that. Whether something is, it's a snapshot. Point being, it's a snapshot. There's concern about this kind of sampling protocol where you go back once every 5 years and what happens in that year. It's part of the reason we now have 4 rounds of our mountaintop data, which I haven't talked about at all, and we haven't even touched it yet because we know it's going to take, or we're about to, but until we had that 4th data point, we didn't feel confident that even having 3 data points would be somehow representative of a trend if we found one, because it could be just so different just based on what happened that year climatically. So we'll see. Yeah, we'll see. It's definitely a concern. Yes, sir.
I think I'm gonna re-ask Bruce's question. By life history, you mean evolution?
I wasn't, meaning evolutionary theories. I was more concerned about the sort of annual life cycles versus perennial life cycle.
Well, then I'll ask a slightly different.
All right.
I mean, it seems that the microclimates are so important, and the variability is at each level is so high that the species don't necessarily have to move upslope. But eventually they will. You're just slowing down the process because those micro variations will eventually move upslope as well, right? And it seems that different species are going to be able to deal with those changes based on their evolutionary history, the ability to adapt, or their past abilities to live in different microclimates at even the same variation. Is that right?
That's
So it's species specific, really, on whether they're going to be able to survive a certain amount, 2% or 2 degrees Celsius.
Gleason is smiling in his grave. Yeah, I think you're dead right. Certainly, my other work with trees shows that, and I think there are kind of 2 things. Species that are better dispersers are really winners. They're showing up as winners, and species that can tolerate a broader range of conditions are also winners. There's Limberpine is a species I do a lot of work on, which has both of those attributes. You find it in the Rockies. You find it from lower tree line to upper tree line, and can tolerate just about anything, and also has a dispersal vector, Clark's nutcracker, and so all over the Great Basin where you find Limberpine, it's thrilled. Climate change is the best thing that's ever happened to it. And it's in direct competition with bristlecone pine, which is considerably less thrilled because it doesn't have either of those attributes. So that's just an example. Certainly there are the grasses have amazing climatic range typically in our systems. Clarimacrantha, Elamus elamoides, they're species you find at valley bottoms and the tops of peaks. They're fine. They're doing just fine. There are other species, some of the Dravas, for example, that have limited dispersal and have a narrow range, I think are more concerned for sure.
So it puts a slight damper on your sort of positive.
Yeah.
A little bit, because diversity is what we care about. And we're still going to lose diversity, even though we're not going to lose species. That's all the species.
Sure, to get philosophical, I think there are multiple things to worry about. Diversity is certainly one of them, but community persistence, I think, is another one. And I don't think we necessarily immediately lose our alpine community. I mean, eventually it gets taken over by trees, and so then you lose. And done some analyses that show that pretty much in the White Mountains, where this research was done, climatically, we're just about out of alpine zone. So yeah, I mean, yeah, sorry, we can feel it too. I mean, I think that's also a reasonable thing to feel.
Well, we had a guy
pessimism.
He gave a talk maybe last year, Bruce, you might remember this. He talked about the ability of plants to adapt quick enough, which I thought was ridiculous.
Adapt evolutionarily. Oh no.
You remember that?
Yeah, and it's interesting because you can present this in a number of different ways, or what's the real outcomes going to be?
Yeah.
And it's really it's as you pointed out, you can come down on either side pretty easily, but you end up sort of cherry picking the right variables to make your case, and we see it in the literature.
Yeah.
But I think that it's the real challenge is to get enough of the factors to feel like you're picking up the variation, and if sensitive enough to see it in some of the methodologies that we're using.
Right.
The Mirren data set is another set. You probably are familiar with that, where they look at non-native species, their distribution along elevational gradients, and yeah, of course, many of those species got to a new continent because they had all kinds of attributes that allow them to go just about any place in the elevation. That's interesting. I mean, in where we were working in Chile, their lodgepole pine introduced there has gone above their tree line.
Right.
Oops.
Yeah. Right.
So there's things like that make it certainly complicated. But you got to be careful to not just take species specific kinds of things and then interpret them in some way. So
right.
It's a challenge.
It is a challenge. I think your point's well taken. I think any optimism is certainly short term regarding microclimate. I think maybe we have one more question, Matt.
So the Sierra Nevada, White Mountains, Sweetwater Mountains, all that.
Yeah.
Quarter there, known to be summer dry. And the Alpine Florida, as a result, of a lot of the surrounding desert, Great Basin desert.
Right.
So, it seems to me as that you're dealing in a unique setting that may not be able to extrapolate other ranges like Rocky Mountains or something like that.
Yeah, I think that's fair. I think that's definitely fair. There's a really gradient going, from there to here, where you get more and more monsoonal activity as you go east. It's part of it. So Europe has done all of these analyses. I showed some of them. It's part of our excitement actually to look at our stuff as being unique and applicable to at least Mediterranean systems, which I think are underrepresented in a lot of the GLORIA sites. And so, yeah, you're right. That's a problem to extrapolating to wetter areas, although I think it's also exciting, and then it provides actually a different kind of system that is probably more represented in the literature for this kind of thing. Good point, though. Yeah, thanks.
Another round of applause.
Oh, thank you.